Hi guys, so today we're going to be taking a look at the evolutionary analysis of lethal amanitas and their biochemical toxins. Here's a little table of contents if you'd like to stop and take a look at it. And we're going to start off with a little taxonomic overview. So the genus Amanita can be broken down into two subgenuses and 7 sections. The subgenus Amanita includes the edible amanitas, and typically a factor that helps to identify the members of this subgenus is that they have enameloid spores. Basically, this means is if they are stained with an iodine-based reagent, such as Meltzer's or Lugal's reagent, they will not absorb that dye. They will not react with it, so their spores will remain pale. In subgenus Lepidella, the spores do absorb and react with the dye, so they get a bluish tint. And the subgenus Lepidella includes the lethal amanitas, which are found within the section Phalloidae. A bit of morphology and adaptations of lethal amanitas. They generally have a caprim that is smooth. They have free gills. They have a collar that can be seen on the body, and they have a universal veil remnant on the bulbous stipe base. The biggest adaptation of the members of the Amanita genus is that they are ectomycorrhizal fungal symbiotes. So basically, they form their hyphae extend and form a network around the plant's roots and cells. And this net that is formed around the cells is called a Hartig net. Doing so allows for a symbiotic exchange of nutrients, so this benefits both the mushroom and the host plant, and it's a very important component to the forest ecosystems, or really any ecosystem for that matter. So the genus Amanita is hard to determine where it came from. Generally, it's accepted that it has a Gondwana origin based on the host species that it's associated with, such as like oaks and pines and beeches. But there are no fossil records of this genus, and details remain unknown. A few methods of dispersal that it could have spread out with is spore migration, continental drift, and anthropogenic influence these days, especially. During the Jurassic period, the split between the two subgenuses happened, and during the Paleocene, we see the emergence of lethal amanitas. A bit of the phylogenetics on these amanitas. Using internal transcribed spacer sequences, scientists were able to create a phylogenetic tree, and in doing so, they were able to find a single origin location of the toxins within the genus. It's interesting, though, that 3 of the members within this genus do not produce the toxins, or rather this section. And it seems to be that that's where the emergence of the toxins really happened. A bit about the lethal molecular compounds. They are cyclopeptides, and they're only about 6 to 10 amino acids long. They're non-essential, so they're called secondary metabolites. And they're usually coded by the MSDIN and POP gene families. However, there's definitely a lot of other gene families that code for cyclopeptides. Many of these peptides remain unknown. There's a complex array of peptide components, and they all consist of different biological functions, and they can be quite varied between species. So amatoxins are the biggest ones. They prohibit RNA and DNA transcription, which makes them solely responsible for fatal human poisonings. In this little picture on the right, you can see RNA polymerase 2 with alpha amanitin in the binding site. I just thought it was a nice representation of it. Phytotoxins are also hepatotoxic, and they bind to F-actin, which prevents cell depolymerization, but they haven't been observed to cause any human deaths. Phytotoxins are similar in structure and activity to phytotoxins, but again, they don't really cause fatal human poisonings. I like to do an honorable mention of ibotenic acid and muscimol, and muscarine, which are toxic but not typically lethal. Basically, ibotenic acid gets converted to muscimol in the GI tract or during cooking, and this sort of mimics GABA, which is usually the reason why members that produce these toxins, such as Amanita muscaria, have psychotropic effects. And muscarine is also an important compound. Again, not toxic, but it mimics acetylcholine, which makes it of interest to neurobiological studies. So, how are these toxins synthesized? Amatoxins and phytotoxins. They're both encoded by the MSDIN gene family, and this gene family is a precursor for the peptides that produce the toxins. And the catalyst which they use is the POP catalyst, so prolyl oligopeptidase. And there's variants of this catalyst, such as POPA and POPB. But basically, amatoxins are bicyclic, and both amatoxins and phytotoxins are ribosomally synthesized. Um, Amatoxins have a sulfoxide bridge, whereas phytotoxins don't. And amatoxins have side chains that have been modified, and this allows for the affinity towards RNA polymerase 2 and 3. An interesting thing that has been discovered is that there's a discontinuous distribution of toxin producing members. So we know that the toxins are produced by the Amanita genus, but they're also produced in the Galerina and Lepiota genera, which are saprobic. So they don't even use the same living strategy as the Amanita genus. They actually are decomposers, so they break down material. And there's three options with which this scenario could have evolved. So there's homology, convergent evolution, and horizontal gene transfer. Homology, which means sharing a deep ancestor, could be possible, especially when considering the families between the similarities between the three. However, it's hard to explain why the intervening taxa have lost this trait despite being subject to similar challenges. Convergent evolution is a good guess, but it's unlikely due to the high similarity in which the proteins are synthesized. So here's a couple of things listed here. I think the biggest one is the fact that the MSDIN and POPB gene families have similar intron and exon locations. So this means that horizontal gene transfer is the most plausible explanation for the three genera to be using similar biosynthetic pathways, and that they're encoded by the same precursor peptide family. And they also use the same catalyst family. So it's hypothesized that the transfer went from Lepiota to Galerina to Amanita, but I'm not super sure how accurate that is. It's just what's being hypothesized. Now, do these toxins offer any selective advantages? Considering that especially alpha amanitin is really toxic to most living cells, it's likely considered to be a defensive strategy. And it's effective against gastropods, nematodes, and flies. Resistance to alpha amanitin has been observed in some organisms. And an interesting thing to point out is that there's no binding affinity to RNA polymerases of eukaryotes or organelles such as mitochondria and chloroplasts. I'm not sure why that is, but it's just something that's been discovered. So there's an interesting relationship between alpha amanitin and Drosophila species. Some species of Mycophagus drosophila, so fruit flies that are like fungus eating, they have displayed considerable resistance to dietary alpha amanitin, And some examples are listed below, especially D-phenyl, though, has been looked at quite a lot. And in lab crosses, alpha amanitin resistance was controlled by 2 genes on the 3rd chromosome in fruit flies. So there's 2 studies that have been done that I could find, which have looked at the resistance to alpha amanitin in fruit flies. They both have shown that non-mycophagous species of flies are extremely sensitive to alpha amanitin, whereas the naturally mycophagous flies were largely unaffected. And it's been hypothesized that this may be beneficial to resistant flies in aiding them with stunting the growth or killing nematodes, gnats, and crane flies, or other parasites that target them. Maybe some alternative reasons that could have led to the production of these compounds. Maybe they offer a competitive advantage against other ectomycorrhizal fungi. For example, Amanita phalloides, which is the death cap, has been introduced to North America in the east and west coasts, and it's been observed by people in some cases to have displaced other naturally occurring ectomycorrhizal fungi, such as chanterelles. So they're becoming invasive. They're replacing native species. And so their toxicity to other fungi has not been studied yet, but it could provide a competitive advantage. The toxins may deter pathogens from plant roots, which they exchange nutrients with. So this might ensure that they can successfully exchange as many nutrients as possible without the plant dying of an infection of any sort. However, the levels of toxins in the mycelium are not known, and it's very hard to even look at that. Also, these toxins have not been shown to aid in the symbiosis relationship, so it's all very debatable. Um, Many of the lethal fungi of this genus lack bright coloring, so I thought maybe there could have been a trade-off that happened between the pigment and the toxin. This is probably unlikely, but it's just a little theory. And maybe these compounds could just be the remnant of an old genetic trait that was retained, but usually natural selection gets rid of unnecessary traits. And there has been a rapid speciation observed within the lethal section of Amanita, so the phalloides section. And especially within the members that are capable of producing the toxins, because as we saw, some members don't produce the toxins, but perhaps toxin production can also drive the rate of speciation. So if there are any questions, feel free to leave them below in the comments. And here are the sources that I used to do the presentation. Thanks for listening.